So as you can see, I moved to Mainz, and where I'm establishing now a small group, but I want to emphasize that all the work that we will show you was done in Janne's Uhle lab, where I spent a really nice time during the last 5 years. So the main interest of my work is the regulation of alternative splicing, and I'm particularly interested in how these auxiliary RNA binding proteins guide and shape somehow the activity of the spliceosome. And what turned out also is that these auxiliary proteins, they don't act individually by binding cis elements and then activating, but they also act in a highly interdependent manner. So there's competition between these factors, there's synergy, and this is somehow, to obtain a global description of this is somehow what I would like to achieve. One core technology, or very powerful technology to study this type of interactions coming from a genome-wide perspective is CLIP, that we heard already about, and I want to introduce you into one variant of this technology that we developed in Janne Uhle's lab, which is called ICLip, and somehow increases the resolution of this genome-wide protein RNA interaction maps that you can achieve. So the outline of this technology, is that by UV irradiating living cells, protein and RNA that are in close contact can be covalently cross-linked, and this then preserves the in vivo interactions for subsequent purification. So then the protein of interest is purified from the cell extracts with an antibody, and to specifically obtain the RNAs that are bound by this protein. And then the protein is removed with proteinase K, and this leaves a short peptide here attached to the RNA at the site of crosslink. And then when you do reverse transcription, a majority of these cDNAs will exactly truncate at the site of the peptide. And then when you prepare a high throughput sequencing libraries out of these cDNAs and sequence into the truncation site, this provides you then very high resolution information about where the protein was sitting on the RNA. We decided to study one protein with that called hnRNPC, and for me this is quite interesting protein because it's highly abundant in the cell, it forms a tetramer, and it's known to nucleate hnRNP particles that have been very early on observed to assemble as somehow beads on a string on nascent RNA, and they've also been referred to as ribonucleosomes, and I think these could be a structure that really globally affect the binding of other RNA binding proteins to pre-mRNA. Now, when we do CLIP with it, what we can observe is that here on this candidate gene that it binds all along the pre-mRNA, where all these peaks here indicate high affinity binding sites of the protein. We also learned from the data that hnRNPC seems to be specifically binding to uridine tracts, and in particular very long uridine tracts. So we observe highest affinity for tracts longer than 9 uridines in a row. And binding of hnRNP seems also to be functionally relevant for alternative splicing. So exons that are repressed by hnRNPC, so this is when you do a knockdown with hnRNP exons that show higher increase, I'm always pressing the wrong button. Then you see an enrichment of hnRNPC upstream of the exon and also a little bit downstream of the exon. And from this and other data, we initially formulated a model for hnRNPC function where binding of hnRNPC to uridine tracts upstream and downstream of an alternative exon would silence this exon, whereas when you would have hnRNPC particles forming within introns, that would enhance the splicing of a downstream alternative exon. Now, this model was very importantly based on one piece of data, and this is shown here. And this was when we aligned all the binding sites of hnRNPC at one position, and then we looked, okay, where's the next binding site what we observed was that there's a peak of binding around 165 nucleotides downstream, and then another peak 300 nucleotides downstream. And we interpreted this as the RNA being wrapped around the particle, and the particle would have would mainly incorporate like 165 or 300 nucleotides, and this would be the footprint of that. But then we came across a review from Jill Ast's group about the evolution of exons, and here we were particularly interested in one part about ALO elements. So we heard about ALO elements before. They cut across the human genome in hundreds of thousands of copies. They are retro transposons, and they consist of two arms, and the arms are separated by an A linker, and then they also have an A tail. And when these ALO elements insert an antisense into a transcript, these A linker and A tail become transcribed as uridine tracts that serve as very good polypyrimidine tracts to be recognized by the splicing machinery. And what turns out is that many of these ALO elements during evolution have been recognized or become recognized by the splicing machinery and became exons. So one idea and one particular important information now regarding the previous slide that I showed you is that the length from here to here is 300 nucleotides, and the length from the first to the second uridine is 165 nucleotides. So this spacing, from there we concluded somehow that the pattern we saw before is because hnRNPC strongly binds to these uridine tracts within ALO elements. And when we looked, okay, where's the clip binding located, we observed, okay, about 25% of all hnRNPC binding goes to ALO elements, strongly underlining that the major function of this protein seems to be recognizing these ALO elements. Now, is this also functional? Yes, so we had also a splicing microarray for hnRNPC knockdown, and we had some ALO exons on it, and almost all of them showed higher inclusion in the hnRNPC knockdown, indicating that hnRNPC would specifically recognize these ALO exons and somehow knock down their inclusion. But then again, there's only a couple of these already exogenized ALOs, but there's these hundreds of thousands of ALOs within introns, and they also have the potential to become exons, and hnRNPC could be involved here. And so again, a study from Jill Ast's lab, they proposed or found that there's about 10,000 ALO elements that have very high probability of being exonized because their splice sites look like proper splice sites, but it was somehow unclear why these don't become exons. And so one possibility was that hnRNPC would bind to all these intronic ALO elements and prevent their recognition to the splicing machinery, and therefore these wouldn't be recognized as exons. And so we wanted to test that, and we decided to choose here for an initial experiment this perfect candidate from this study here, the ICGEB that you heard before, and we did PCR, RT-PCR on it. So in the control there's no inclusion of the ALO exon, but then in the hnRNPC knockdown we observe a very slight inclusion of this ALO element within the ICGEB gene. Even though this inclusion was very faint, maybe 1%, but we were at this point convinced that hnRNPC could have a major role in preventing the exonization of these ALO elements, and therefore we decided to make an RNA-seq analysis of hnRNPC knockdown cells, because in contrast to the microarray, this allows us to de novo annotate the transcriptome and to identify exons that have never been described before. And in fact, we find hundreds of exogenizing ALO elements And this is one example of this. So this up here is wild-type RNA-seq, nice peaks on all the exons. And then here, this is knockdown. And here we see coming out of the middle of nowhere, where an ALU element is located, a very strong peak indicating that this region has now been included into the mature transcript as an exon. So what do we have at this point? So we see hundreds of ALU elements coming up in the coming up as exons in the hnRNP knockdown. And we know that hnRNP binds to the uridine tracts within the aluments, indicating that binding to these uridine tracts is the reason why these ALU elements are kept silent. And to look a little bit more into the mechanism, so you can see this here again, the ALU element with the two uridine tracts where the hnRNP binds to. And as I pointed out before, these uridine tracts here act as a polypyrimidine tract signal for these exons to be recognized. And when we look here at the schematic, the polypyrimidine tract, as you also heard this morning, is recognized by the very important splicing factor U2AF65. And one possibility would be now that binding of hnRNP to these uridine tracts would prevent their recognition through U2AF65, and this would be the mechanism of preventing the inclusion of these cryptic exons. And to test this, we again made clip experiments, and this time for U2AF65. And these turned out to be really nicely, I think, because we observed for almost all the exons, very strong peak of U2AF65 binding immediately at the 3' splice site, absolutely consistent with years of understanding of U2AF65. Now, in order to understand whether hnRNP affects U2AF65 binding, we repeated these U2AF65 IClip experiments in hnRNP knockdown cell lines, so in the absence of hnRNP. And first, globally, we don't see a lot of difference, so all the peaks at the constitutive exon seem to be unchanged. But then when we zoom in here on this ALU-derived exon in this gene, what we see is a different picture. So this is now nucleotide resolution here. We have on top a very nice footprint of hnRNP binding on this very long uridine tract in front of this ALU exon. In the wild type, relatively little U2AF65 binding, but then in the knockdown of hnRNP, we observe a strong increase in U2AF65 binding, really indicating that under normal conditions, hnRNP sits there, U2AF65 cannot come, and then when hnRNP is gone, U2AF65 goes in and activates splicing of this exon. Okay, so we don't see this only on individual genes, but or in individual ALU exons, but genome-wide, or on several hundreds of ALU exons. So now here, each of these lines in this heat map represents one individual ALU exon, where the dotted line is the 3' splice site. And then these blue points here indicate hnRNP cross-linking. So the ALU exons seem almost all of them seem to have very strong binding of hnRNP upstream of the 3' splice site. And this is absent when looking at a subset here of control exons. And then when we look at U2AF65, there's relatively little U2AF65 cross-linking. But when we look now in the hnRNP knockdown, this U2AF65 binding strongly increases in 3 minutes. Okay. So we also, one question was, okay, so how does hnRNP now differentiate between constitutive exons where it seems not to affect hnRNP between these ALU exons? And the answer probably lies in the different sequence specificity of the two proteins. So hnRNP really prefers these long uridine tracts, whereas U2AF65 likes uridines but can also tolerate cytidines. And we decided to study this also a bit in vitro. And what we did was we took short RNA oligonucleotides here, contiguous uridine tract and incubated it with constant amounts U2AF65, and then we added in increasing concentrations of hnRNPC, and then when you look at the crosslink, so this is radioactively labeled, and then crosslink to the protein, what you then see on the RNA level is that with increasing hnRNPC concentration, U2AF65 binding decreases, whereas hnRNPC crosslinking increases, recapitulating this competition on this short oligonucleotide. When we now have a control oligo where we put in these cytidines interrupting this contiguous uridine tract, see a completely different picture. So U2AF65 binding is not affected by addition of hnRNPC, and hnRNPC doesn't gain access. So by changing the sequence composition of this little oligo, we can shift the balance between the competition of these two proteins. Okay, so just one last slide. Somehow to underline that this is a very important mechanism. Somehow we were contacted at some point from the lab of Xiaobing, and they are interested in the double-stranded break repair pathway, and they found somehow a connection between hnRNPC and this pathway. And what they did was they knocked down hnRNPC, and then they saw decrease also in the proteins of 4 very important factors: BRCA1, BRCA2, RAD51, and BRIP1. And they asked us, okay, could you explain what is the reason for that? And we looked into the RNAs, and what we observed was that in in all these 4 RNAs, we observed ALO exons coming up in the knockdown of hnRNPC, and this then leads to premature termination of the truncated proteins and so on. Okay. Okay, and then with this I would like to finish. Would like to thank Jerne, first of all. Really nice time in his lab. When you get the chance, join him. Then of course Moitza, we worked together on various experimental aspects of this project, and then all the computational work was done by Kati Sarnak in Nick Laskamp's lab. And yeah, thanks for attention.